Welcome to today's lecture. This is in continuation about the meat quality. In the previous lecture, we have discussed about the fresh meat quality part 1. Today is the fresh meat quality part 2. Today we will discuss about color, texture and tenderness of fresh meat. So in color, we will understand what are the attributes of color and what are the chemical aspect, the meat pigment and how the chemical changes happen and color changes and what are the factors behind it. And then we will understand what is the texture and factors affecting the texture of meat. Then we will understand about the tenderness, what are the different aspects of tenderness, and there are pre-slaughter and post-slaughter factors in tenderness. So all these things we are going to discuss slowly in this lecture. So we are going to discuss about the color of meat. Before that, first let us understand a basic aspect of color, that is the attributes of color. Color has got three aspects or attributes, that is hue, chroma and value. Hue is the perception, the first perception about the color. It describes that which one normally thinks of a color, that is yellow, blue, green or red. The second aspect is the chroma, that is about the purity or intensity of that perception of color, and it describes the intensity of fundamental color with respect to amount of white light that is mixed with it. And the third attribute is the value, that is about the brightness of that color. So this is an indication of overall light reflectance or brightness of the color. So these are the three basic aspect of color which determines the overall impression or perception of the color of anything. Here let us first understand what is the color of different meat. So this I have discussed in the basic class in undergraduate level. Now whatever we are discussing, this is for postgraduate level. So the color of beef, in general it is described as bright cherry red. In case of veal, that is from the young calf, it is brownish pink. Poultry, grey white to dull red. Pork, it is greyish pink. Mutton, it is light red to brick red. Mutton means it is from the sheep, as you know. And gibbon is from goat, that is light red to brick red. And in case of horse, though it is not common in India, it is sometimes used in European countries, that is dark red. So these are the normal color of different type of meat. In the previous slide, we have told about the color of different type of meat, but this color is directly related to amount of myoglobin. So in a previous lecture for UG, there I have given the myoglobin content in different species meat or muscle, and we can see that the myoglobin content is very high in case of horse, that's why it is very dark, whereas in case of poultry or rabbit meat, myoglobin content is very less, so it is light. So here we will see that factors affecting the myoglobin content in meat. Within the species, like between the sex. So sex has influence on myoglobin content of meat. So bull has more myoglobin than cow, so the meat from male will be little more darker color than the female. Within the species, age. So myoglobin content increases with age due to the more activity, growth and other things. So that is why in case of veal, that is the meat from young calf, will be light color than that of beef. Then type of muscle. So between muscle also there is a color difference due to there is a difference in muscle fiber and their function. So some muscle has got more red fiber, some muscle have less red fiber. So that makes a difference in color. Similarly, the function or activity. So those muscle which are high active, they will have more myoglobin and dark in color. And similarly, more working. Some animals who are working, some muscle will be more working. So those muscle will be having more myoglobin and dark. And same way the exercise, the increases the myoglobin content. Therefore, myoglobin content of free range animals are high than the confined animals. Animals which are free, more working or running, they will have more myoglobin and color will be dark. Now first we will understand about meat pigment. The major pigment for meat color is myoglobin, that is also we can say the muscle pigment, which is mainly the responsible for color of muscle or color of meat. In well-bled carcass, myoglobin constitutes 80 to 90% of the total pigment, followed by hemoglobin. Because of the bleeding, the hemoglobin will go with the blood, so the major color comes from the myoglobin. There are other pigments like catalase and cytochrome enzyme, but their presence are not felt in the color because they are in a very minor quantity. So the major muscle pigment or meat color is due to myoglobin. Now here we will understand the how color varies or how the color depends on what. The appearance of meat surface mainly depends not only the quantity of myoglobin present but also the other factors. Of course, the quantity of myoglobin is one of the most important, and the other factors are like type of myoglobin molecule, its chemical state. The chemical state of myoglobin will vary depending on the situation or atmosphere, which we are going to discuss in details, and the chemical and physical condition of the other components of the meat. Like along with meat there is fat. So fat in what condition, that matters. So if fat get oxidation, that will affect the myoglobin and also the color. Now here we will understand little more details about the myoglobin and its status. So it can anytime get oxidized or reduced. So that oxidation or reduction status has an important correlation with the color development of muscle or meat color. So in myoglobin structure, if we see, there are two parts. One is the globin portion, which is a protein. Another is a non-protein portion, that is the heme ring. So this heme ring is a protoporphyrin ring. At the center there is a ion. So the oxidative status of ion is directly responsible for color of meat. So this heme ring portion contains ion, and the oxidation state of ion within this heme ring partially determines the color of meat. And this is what is very important in case of meat color and meat science. We are going to see again about it. Here little more we will understand about the chemical state of heme, that is mainly due to the ion. The myoglobin can react with different compounds and ions including water and oxygen when the ion within the heme ring is in reduced state. So when it is in reduced state, we call it ferrous state, and at this state it can react with other compounds and ions, and that plays important role. So when the ion is in oxidized state, then we call it ferric state, and it cannot combine with other molecules or other oxygen. So this makes the major difference. So it is important to maintain the reducing condition of the pigment in fresh meat because oxygen reacts with reduced ion of myoglobin to provide desirable color of fresh meat. The desirable color of fresh meat is the bright red color. That color comes with the combination of ion with oxygen, that is we call oxymyoglobin. So that is possible only when the ion is in reduced state. When it is in oxidized state, then it is met myoglobin. At this stage it cannot bind with oxygen, so we cannot get the bright red color or desirable color. Here we can see the change of oxidative status of ion in the myoglobin, that is in the heme pigment. We can see three different states is possible. One is deoxymyoglobin, that is in a reduced state. So the ion is in ferrous state here and the color will be purple, and when it binds with oxygen it forms oxymyoglobin, but it is still in reduced state, but the color will be bright. It is by combination with oxygen, not oxidized. But this oxymyoglobin or deoxymyoglobin both can get oxidized. There the pigment become ferric and then it becomes brown color, and that we call met myoglobin. So there is a interchange between these three. Deoxymyoglobin or reduced form can also get oxidized and form met myoglobin, whereas the oxymyoglobin also due to certain condition get oxidized and form met myoglobin. So there is a interchange depending on the surrounding condition and other factors. The pigment remains in three different states, and accordingly the color changes like purple, bright red and brown. Here once again we will see the state of pigment and color, which I have already explained. when freshly cut meat is exposed to air, the reduced pigments will react with oxygen and form relatively stable pigment called oxymyoglobin. This formation of oxymyoglobin in fresh meat is responsible for its bright red color, which is otherwise termed as bloom. So this is the desirable color due to the formation of oxymyoglobin, and this is the bright red color which is preferred for the fresh meat, and that is called as bloom. Whereas oxidized form of myoglobin, as I told, it is metmyoglobin, and that imparts brown color which is not attractive and not desirable. So this is due to the oxidation of ion or ion in the heme and making it metmyoglobin. Here once again we can see that fresh meat color triangle from deoxymyoglobin, ferrostate. Sometimes it can be carboxymyoglobin when there is no oxygen and carbon monoxide is added. But when there is no air or anaerobic condition, it may lead to metmyoglobin formation. It happens due to other condition also, and the deoxymyoglobin to oxymyoglobin, which I have already explained earlier. So all these conversion are also affected by pH and temperature. pH and temperature plays very important role for this oxidation or reduction, which we will see again. Here we can see the relation of pH and color of meat. We have discussed earlier in many lectures that due to the post-mortem changes, there will be fast decline of pH and leading to a ultimate pH. But the rate of decline has an important role. When it is very fast and very quick fall of pH, that leads to pale color, which I have explained in the previous lecture, the reason. And when it is very slow and not going down much to the ultimate pH, then we get dark color, whereas the middle one is the normal. So here, due to the pH, the water holding capacity changes, and in case of the top dark one, here much of the color it is absorbed, so it looks dark, whereas in the down, in case of pale, the much of the color is reflected, so it looks pale. So all these things we have discussed in the previous lecture. Here once again we will discuss about the formation of metmyoglobin. Metmyoglobin is formed when small quantities of oxygen are present, as in case of partial vacuum or a sealed semi-permeable package. So when we do a vacuum packaging in fresh meat, so there will be no air, then that lead to oxidation and metmyoglobin formation. Or sometime if we have a packaging material which is not permeable to oxygen, again it may lead to slowly formation of metmyoglobin. So formation of metmyoglobin in fresh meat must be avoided because consumers generally relate this brown color as if it is stored meat or old meat, and they don't like it or reject it. So that's why the supply of oxygen should be there and oxymyoglobin formation should be there. Under normal atmospheric condition, oxymyoglobin, that is the oxygenated pigment, is stable and not easily oxidized to metmyoglobin. So that gives the bright red color which we call bloom. So here we will talk about the formation of oxymyoglobin. Formation of oxymyoglobin in meat is spontaneous when it is exposed to air. So there should be sufficient air which will have oxygen, and if the heme pigment or ion pigment is in reduced state, automatically it will bind with oxygen and form oxymyoglobin. But its stability depends on continuous supply of oxygen because enzymes involved in oxidative metabolism rapidly use the available oxygen in the muscle tissue. So there has to be supply of oxygen, otherwise if it is in a tight package without any air permeability, then slowly the oxygen will be used by other enzymes and other metabolic process. So later there will be no oxygen. So after some time or during storage, the oxygen will not be there and it becomes metmyoglobin and it becomes brown red. So the pH and temperature of the tissue or the muscle has an important role here. If the pH and temperature is high, then this enzymatic activity and oxidative metabolism will be more, so the oxygen will be utilized quickly and then there will be no oxygen supply. So depending on this, the pH should be low, pH should be maintained at a lower temperature, meat should be kept so that the enzyme activity can be reduced. Here we will see that temperature and color has a relation even with pH, as I mentioned already. So maintaining the temperature of meat near freezing point minimizes the rate of enzyme activity and oxygen utilization, and thus helps to maintain a bright red color for maximum possible time. So if the temperature is low, all the enzyme activity, metabolic activity will be less, so oxygen supply will remain for long time. The bright color can be maintained long time. Usual color development in fresh meat include pale, that is the pale soft exudative condition, that's a abnormal condition, and another is dark or dark farm dry meat. These two we have already discussed earlier lecture also. So this happens already due to the improper anaerobic glycolysis, which I have mentioned in the pH decline, post-mortem pH decline. Here we can see the color changes. At the middle it is normal, at the left it is DFD, and at the right it is PAC. Similarly, in the bottom diagram we can see that pH 5.5, and when it is increasing to up to 7, the color is becoming more and more dark. So ideal point is 5.5. Now we will discuss about the next fresh meat quality parameter, that is the texture. Texture and tenderness are the most important eating quality parameters, but texture is expressed from fresh meat more commonly. So texture as seen by the eye is a function of the size of the bundles of muscle fibers, which are covered and separated from each other by perimysium. So we have discussed in details in a undergraduate lecture about the muscle structure. The muscle structure has got three different kind of connective tissue covering, that is the epimysium, perimysium and endomysium. Epimysium is covering the whole muscle, whereas perimysium covering the bundles of fibers, and the innermost endomysium covering the each muscle fiber. Now here we are concerned about the bundles. So these bundle size determines the texture of the muscle. So if it is called coarse grained muscle, means that has got large bundles, the diameter of the bundles are more. And when we think it is a fine grained muscle, that means it has got small bundles. So that gives the coarseness or fineness of the muscle texture. Here we can see the factors affecting the texture of meat. As usual, it is the species that plays an important role in different texture. Then within the species, different breed has a variation. Within the breed, different age. The younger age and the older age has a difference in the texture of muscle. Then sex, that is the difference between the male and female. Frame size of the animal, that is overall size or conformation, that makes a difference in the texture. And also type of muscle or between the muscle, due to the activity or less activity, there can be a difference in the texture. So all these things briefly we are going to discuss. Now we will discuss briefly different factors affecting the texture of meat. The first one is the species. Texture of meat varies with species of the meat animal. That is, meat of cattle has coarser texture than meat of sheep or pig. So it is the difference. In case of cattle or the beef, the texture will be more coarse, whereas in case of mutton or pork, the texture is not so coarse. Now about the breed. So within a species, between the breed, there can be difference. Breed influences the meat texture to a varying degree. Meat from Hampshire, Duroc and Landrace pig is coarser than that from Berkshire or Tamworth. So these are different breed of pig, but some pig are having coarse texture, whereas others are having fine texture of the meat. Then another one is the age. As I have mentioned earlier, the coarseness of the texture increases with the advancement of age of the animal. So in the young animal it is little bit fine, and as it grows more and more older, the texture becomes coarse. Now in continuation about the factors affecting texture of meat, the another one is sex. So muscles of the male animal has greater coarseness as compared to muscles of female animal. So the meat from male and female, there will be difference. The meat from male will be more coarse in texture than from the female. It is also partially maybe due to the more muscular activity. Then frame size of the animal. The frame size of the animal also affects the texture of the meat. The coarseness of texture is greater in the muscles from the animals of large frame. So if the animal is of large size, naturally it is expected to have coarse texture in the meat. And then type of muscle. The muscles such as biceps femoris or semitendinosus of the hind leg, which perform vigorously in live animal, are coarser in texture than the infrequently used muscles such as psoas major of the loin. So I mentioned in the previous one, those muscles which are more active, they will have more coarse texture, whereas some muscle which remain inside at the front portion, psoas major, they have very little activity, so they may be little bit less coarse or fine in texture. Now we will discuss about tenderness. This is actually to be perceived during eating, so this comes under eating quality, but it is directly related to the fresh meat and it plays very important role in determining the quality of meat. The overall impression of tenderness to the palate includes texture and involves three aspects. So texture is directly connected with the tenderness. The first aspect is initial ease of penetration of meat by the teeth. Then second is ease with which meat breaks into fragments. And the third is the amount of residue remaining after chewing. So these three aspects, how easily the penetration happens by the teeth to the meat. Second is how easily the meat breaks. Third is how much residue is remaining. And another is sometime number of chewing required before swallowing. So these things decides or indirectly reflects about the tenderness of meat. So we will see different aspect of it. So now we will see different components of tenderness. The sensation of tenderness has several components of varying importance, and perception of tenderness by human is very difficult by scientific instrumentation. So it is not easy to correlate the human perception with the machine measurement. So the measurement of tenderness also done by instruments. That's why we are discussing it in this topic in the fresh meat quality or fresh meat properties. The major components of meat that contribute to the tenderness are three aspects. One is the connective tissue, which I told, that is the three layer of connective tissue on the muscle, the epimysium, perimysium and endomysium. The second is muscle fiber structure, the structure of the muscle fiber, how it is, the amount of muscle bundle size and that coarseness, all this thing has a relation with the tenderness. And the adipose tissue, that is the deposition of fat. So earlier we have discussed that intramuscular fat deposition, which we also call marbling. So that has a direct relation with the tenderness. So these things again we are going to discuss on different angles. Here first we will discuss about the connective tissue and tenderness. The amount and nature of connective tissue protein, that is mostly collagen in muscle, determines the toughness of meat. So collagen and its quantity and nature has a direct relation with the tenderness, and this background toughness of meat is contributed by connective tissue present in endomysium, perimysium and epimysium of muscle tissue. I mentioned that this three layer covering the muscle has got mostly collagen. So this collagen has a direct relation with the tenderness, and that is called background toughness of meat. Now in young animal, although amount of connective tissue is comparatively more than that of mature animal, the background toughness is less in meat from young animal compared to the old. So this is because as the age increases, the nature of collagen changes, and more it forms the cross linkage and it becomes more tough or more insoluble. The collagen becomes more insoluble with the formation of more cross linkage. So when after cooking, in case of young animal, this collagen easily get converted to gelatin, whereas in case of old animal it is not so easy. So collagen plays an important role, that is from the connective tissue, in the tenderness. Here once again we will see about the collagen and tenderness, as I have mentioned in the previous one, that this old animal will have more mature collagen due to the formation of cross links, and that easily won't get converted to gelatin, so they are more heat stable. Whereas in case of young animal or the animals which are grown very rapid, like all kind of broiler animals, there the collagen is more heat labile and easily get converted to gelatin during cooking, so that gives a more tender meat. The degree of tenderness of muscles is determined partially by post-rigor contraction of the muscle. So this is another aspect which we have discussed earlier. If the rigor is not properly resolved, then there will be a little bit of shortening and that gives it toughness. So that's a different aspect. As I mentioned, the loss of tenderness occurring in the first few hours post-mortem has been known as ectomyosin toughening. So that is the permanent ectomyosin formation leading to formation of rigor. So this rigor and tenderness is also related. Until the rigor is over, muscle will be tough, or just before rigor it may be little tender. So during this shortening and cross bridge formation period, the tenderness and water holding capacity will be minimum, and at this stage the protein interaction making binding sites unavailable for water binding. So water binding is less, so water holding capacity is less, it will be less tender. So the tenderness and water holding capacity are also correlated. Now we will discuss about tenderness by aging process. So we know the after rigor mortis or after slaughter, the animal will be hold for more time for aging, that is after the rigor mortis. So during this period, due to enzyme action, there will be progressive degradation and increased fragmentation of myofibrillar proteins and jet discs, resulting in redistribution of ions and shifts in water binding activity, causing an improvement in tenderness and water holding capacity. So during this process, due to the enzymatic, particularly catheptic enzymes acts on the myofibrillar proteins and also jet discs, so many proteins will be broken and there will be further tenderness is possible, and also by improving the water holding capacity. The other aspect is intramuscular fat, which I have already discussed, that is the marbling. Indirectly it influences the tenderness. So this is mainly because some lipids which act as lubricant during mastication of meat, thus improving apparent tenderness and easing the process of swallowing. So this intramuscular fat, which we are telling marbling, after cooking it remains inside semi-solid inside the muscle pieces, and when we bite and eat, that lipid semi-solid helps in better mastication and easy swallowing and indirectly improves the tenderness. Now we will see the factors affecting tenderness. It can be in two categories like intrinsic factors and extrinsic factors, or sometimes we can say pre-slaughter factors and post-slaughter factors, that I am coming later. In the intrinsic factors, there are some factors which are less important like fiber type, connective tissue, muscle and adipose ratio, and the more important structure of myofibril, amount of glycogen and proteolytic activities. Then there is extrinsic factors in the right side, where first is the live animal, where it is about the animal breed, animal handling and slaughter, and nutrition and climatic conditions. So we can say these are pre-slaughter factors. And then there are slaughter related to slaughter animals. Those are some factors like time, temperature, handling during the slaughter, etc. As I have already mentioned, the factors affecting tenderness of meat can also be classified into other category like pre-slaughter factors and post-slaughter factors. So we will discuss in details about the pre-slaughter factors related to animals and the post-slaughter factors related to practices and other atmospheric and environmental conditions. In the pre-slaughter factors, we have the species of the animal, age of the animal, sex of the animal, and different muscle. So we have already seen similar differences in case of texture. So same way they are having the influence. We will discuss little more next. Now we will discuss the pre-slaughter factors one by one related to tenderness. First is the species. The tenderness is heritable to an extent over 60%, and different species meat has different degree of tenderness. So due to the species there is a difference, like pork is more tender than the beef meat. Then about the age. Tenderness of meat decreases with the advancement of age of the animal, which we have already discussed earlier. It is mainly due to the changes in connective tissue and also quantity of connective tissue and the cross linkage in the collagen. So that makes the meat more tough in case of old animals. So in continuation of pre-slaughter factors, the sex, that is the male and female, tenderness of meat from female animal differs from that of male animal. We have seen it earlier in case of texture also. The male muscle will be more active and there will be more coarse texture and there will be more connective tissue, leading to more tough meat from male. Then about the muscle. So between muscle there is difference. So tenderness of meat varies from muscle to muscle. Psoas major is more tender than longissimus dorsi. That is because of the activity of the muscle. And then muscle which contain more elastin is tougher, and which contains intramuscular fat is more tender. So some muscle has got more intramuscular fat, that is the marbling, so they appears to be more tender. Now we will see the post-slaughter factors affecting the tenderness. So in this we have like post-mortem changes because of the temperature and other aspect. The post-mortem changes will be different. Then conditioning, that is also called aging, that is the holding the carcass for certain period of time for proper glycolytic changes and aging changes. Then chilling, so how quickly we change or how long we hold it at a higher temperature, that matters. Then cooking of course has a direct impact on the tenderness. Then artificial tenderizer, different kind of enzymes especially from plant origin are used as a tenderizer. And then electrical stimulation. Nowadays many times electrical stimulation is used for quick chilling, and that helps in better tenderness also. So briefly we will discuss all these factors. We are discussing the post-slaughter factors for tenderness. The first one is post-mortem changes. When post-mortem pH falls slowly, tenderness will be increased. Toughness of the meat increases with the greater degree of interdigitation of ectomyosin during rigor mortis. So if the pH fall is slow, then the ultimate pH achieved is very late, and that leads to the improper tenderness. The other conditioning or aging changes also will not be ideal. So this conditioning is about holding the meat at a above freezing point for a certain period of time, so that there will be desirable changes from the enzymes, especially the catheptic enzymes. So when the meat is conditioned, stored at chilled temperature for several days sometime, and then the tenderness improves, even the flavor improves. Then about the chilling. If the meat is chilled pre-rigor, then cold shortening, that is pre-rigor shortening, decreases the tenderness of meat. So if we keep the meat after slaughter immediately at chilling, so before the rigor mortis is over, that leads to a cold shortening, and that will give the toughness and the tenderness will be less. When the meat is frozen pre-rigor and thawed rapidly, it undergoes thaw rigor, which again makes the meat tougher. So before rigor mortis is over, if we frozen it, then during thawing there will be a shortening, or that is called thaw rigor, and that causes the meat tough. In continuation about post-slaughter factors on tenderness, so cooking is very important. Cooking helps in increasing the tenderness in general by converting the collagen of meat into gelatin. So I have mentioned earlier, the connective tissue, mainly the collagen, remains in the muscle at 3 layers. So during cooking they get converted easily into gelatin, but in case of meat from older animal, it won't get easily converted to gelatin. So that becomes little tough. Sometime in case of old meat, it is advised to cook with a pressure. So that helps in tenderization. But in general, myofibrillar protein becomes little hard, so that is not easily contributing towards the tenderness by cooking. Then another factor is artificial tenderization. So that is a common practice to apply different kind of plant origin enzymes. So these type of enzymes having the proteolytic enzymes and other mineral salts acid also sometimes is used for marination, and all this thing helps in tenderness artificially. And then electrical stimulation, this is a modern industrial practice especially for quick chilling. So electrical stimulation is given so that the rigor mortis resolved very quickly, and rigor mortis gets over quickly, then it can be kept in chilling for quick chilling. So in this case the electrical stimulation also helps in tenderness. So now we are at the end of today's lecture. Let me summarize. Today first we have discussed about the color, what are the different aspect of color, the chemical aspect, the meat pigment, color of different meat, how the meat pigment changes depending on the pH and oxygen availability, etc., and how the different undesirable color develops, we have discussed. Then we have discussed about the texture, that is more pertaining to raw meat, different aspect of texture and what are the factors affecting the texture. And finally we have discussed about tenderness. So tenderness, though it will be perceived only during eating, so it's a eating quality parameter, but we can measure the tenderness in the raw meat also by using the instruments. And that we have discussed what are the factors of tenderness, like there are pre-slaughter factors and post-slaughter factors. So all these factors we have discussed briefly. So altogether we have discussed about the fresh meat quality. It is in the second part. So one more lecture in this continuation I will have, that is about overall factors affecting the fresh meat quality. There are different kind of factors affecting the fresh meat quality. So that we will have in the next lecture. Thank you.